In various stages of vaccine development and manufacturing, some vaccines have used cells originally isolated from fetal tissue, often referred to as fetal cells or fetal cell lines, some of which were originally derived from an aborted fetus. The use of fetal cell lines is a very sensitive and important topic within some faith communities and among individuals with concerns about the ethics of using materials derived in this way. So let's take a deeper look at how and why fetal cells are used in vaccine development. In the mid-20th century, scientists at the Wistar Institute in Philadelphia were focused on creating cell lines, groups of identical cells grown from a single cell, that would grow continuously in the lab. Cancerous cells grew well and were an option, but researchers wanted non-cancer cells because they were more predictable and better for studying normal human biology. Some researchers had used monkey kidney cells or rodent tissue, but these did not grow well in the lab and had to be prepared fresh for each use. The use of animal cells was cumbersome, costly, and required a large number of animals, making it difficult to consistently assure the quality of cell lines. In his effort to create a continuously growing cell line, Dr. Leonard Hayflick chose to work with human cells. However, Hayflick was worried that cells from people or animals might inadvertently be contaminated, and this would affect his results. He reasoned that a fetus in the womb is protected from most outside pathogens, and fetal cells would offer the cleanest source of cells for his experiments. His concerns about contamination became a reality. In the 1950s and 1960s, the polio virus used to make polio vaccines was grown in monkey kidney cell lines, and some of the monkeys were infected with simian virus 40, also known as SV40. This meant that some of the vaccines also contained SV40. As soon as scientists became aware of the contamination, they actively tried to determine whether these viruses in the polio vaccine caused harm. Luckily, they didn't, but that was not known at the time. In the end, the SV40 contaminated polio vaccine was a major driving factor for Hayflick and his colleagues to develop a new cell line. Dr. Hayflick developed a fetal cell strain called WI38. The strain was developed using cells from the lung tissue of a 3-month gestation fetus taken from an elective abortion in a healthy Swedish woman in 1963. Dr. Hayflick obtained the cells, grew and multiplied them in the lab, and preserved them for future use. WI38 cells were the first and most extensively studied normal cell line to be grown in virtually unlimited quantities. Hayflick found that many viruses grew well in WI38, paving the way for the cell line to be used for vaccine production. WI38 had other advantages too. It was derived from a single donor, was free from contaminating viruses, and could be frozen for indefinite periods of time. His success with developing WI38 was a critical innovation in the development of human virus vaccines. Dr. Hayflick distributed WI38 for free to vaccine manufacturers worldwide. The first vaccine made using fetal cell lines was an adenovirus vaccine for military use. Other vaccines followed, including the rubella vaccine developed by Dr. Hayflick's colleague at the Wistar Institute, Dr. Stanley Plotkin. Rubella is a viral infection that typically causes a mild illness in children, but can be very serious for pregnant women and their unborn babies. Congenital rubella syndrome is a condition that occurs when a fetus is infected with the rubella virus in the womb. It can cause a range of birth defects, including but not limited to deafness, heart disease, encephalitis, eye issues, and pneumonia. At the height of a rubella epidemic in the mid-1960s, Dr. Plotkin calculated that 1% of all births at Philadelphia General Hospital were affected by congenital rubella syndrome. In some cases, women who were infected with rubella while pregnant terminated their pregnancies due to the risk of severe birth defects caused by congenital rubella syndrome. Following one such abortion, the fetus was sent to Plotkin at the laboratory he had devoted to rubella research. When testing the kidney from the fetus, Plotkin found and isolated the rubella virus, a crucial step in vaccine production. The WI38 cell line was chosen for rubella vaccine research for a few reasons. First, the virus grew well in fetal cells. We know that babies born to women infected with rubella early in their pregnancy would often be born blind, deaf, autistic, mentally disabled, or with heart defects. Second, Plotkin chose to use WI38 for the same reason that spurred Dr. Hayflick to develop this new cell line in the first place. It was clear of contamination. Dr. Plotkin wanted to avoid the potential risks seen with the contaminated polio vaccines, and his choice to use WI38 circumvented this concern. In an attempt to weaken the virus and make a vaccine, Dr. Plotkin grew the rubella virus in WI38 cells at 86 degrees Fahrenheit, or 30 degrees Celsius. After the virus had been grown through the cells 25 times at the lower temperature, it was no longer able to replicate well at normal body temperature, or enough to cause illness in a living person, but it was still able to stimulate a protective immune response. The rubella vaccine developed with WI38 is still used throughout much of the world today as part of the combined measles, mumps, and rubella vaccine. Dr. Plotkin faced scrutiny from his colleagues for developing the vaccine using fetal cells, which had many ethical considerations. However, Dr. Plotkin had watched parents struggle with the notion that their unborn child might have disabilities that would shape their entire lives. This left him to believe that the decision ultimately was the right one. Dr. Plotkin, in response to his use of WI38, stated, "From my personal point of view, having seen the suffering of families with congenital rubella syndrome infants, I consider that what was done is 100% moral." In 1963 and 1964, before the vaccine was available, rubella swept through the U.S., infecting about 12.5 million people and causing congenital rubella syndrome in approximately 20,000 babies. The vaccine was licensed in 1969. By 2005, rubella was eliminated from the U.S. Over time, scientists and government officials accepted that the use of fetal cell lines in development of vaccines was safe and effective. The WI38 fetal cell line has been used to develop life-saving vaccines. In fact, researchers have estimated the number of lives saved and morbidity reduction associated with the discovery of the first human cell strain used for the production of licensed human virus vaccines using WI38. The total number of cases of polio, measles, mumps, rubella, varicella, adenovirus, rabies, and hepatitis A averted or treated with WI38-related vaccines was 198 million in the U.S. and 4.5 billion globally. The total number of deaths averted from these same diseases was approximately 450,000 in the U.S. and 10.3 million globally. Which fetal cell lines are used for vaccine development and production today? The fetal cell lines that have been and are currently being used to develop vaccines are from the following sources: WI38, a cell line we've already discussed; MRC5, a cell line made up of fibroblasts isolated from the lung tissue of a 14-week-old male embryo in 1966 from an elective abortion; HEK293, a kidney cell that was isolated from a fetus in 1973 from undisclosed origin from either a spontaneous miscarriage or an elective abortion; PERC6, a retinal cell line that was isolated from an aborted fetus in 1985. Any vaccine that relies on these historic cell lines will not require nor solicit new abortions. In what ways are fetal cells used in vaccine development and production? Fetal cell lines can be used at different stages of vaccine development and production. The first is design and development. Fetal cell lines can be used for conceptualization, introductory experiments, and to provide specification for how the vaccine will be constructed and produced. The second is production. Fetal cell lines can be used to manufacture the final vaccine or parts of the vaccine that will be given to people. The last is confirmatory lab tests on production. Fetal cell lines can be used to analyze the quality of the vaccine, including assessing nucleic acid or protein sequences, protein conformations, and antibody reactivity, among other aspects of the final vaccine product. Which vaccines use fetal cell lines in their development or production process? As you can see, vaccines for varicella, rubella, hepatitis A, rabies, and some COVID-19 vaccines are all made by growing the viruses in fetal cells. COVID-19 vaccines made by Pfizer and Moderna used fetal cell lines for some confirmatory lab tests early in their development, but these vaccines do not use fetal cells to produce the vaccine. Why are fetal cells preferred over other cell types? As mentioned previously, in order to grow a virus for a vaccine, it needs a cell to grow in. There are human viruses that don't grow well in animal cells, so we need to use human cells to grow the viruses. It's also important that the cell line is free of contamination with unwanted viruses, as mentioned in the example of contaminated cell lines earlier in the video. Fetal cells are isolated from a sterile environment, so the cells are much less likely to be infected with other viruses compared to cells obtained from people or animals. We also know that almost all cells die after they've divided a certain number of times. This is called the Hayflick limit, and it will be touched on towards the end of this video. For most cell lines, including fetal cells, the limit is around 50 divisions. Because fetal cells have not divided as many times as other cell types, they can be used longer. Lastly, fetal cells can be maintained at low temperatures, and this allows scientists to continue to use cell lines from decades ago. Do vaccines contain parts of fetuses or fetal cells? Vaccines do not contain parts of fetuses or fetal cells. We'll explain how viruses are grown in the lab to help you better understand why vaccines do not contain fetuses or fetal cells. In order for viruses to grow in the lab, they need a cell to grow in. Scientists start by making single-cell suspensions, where the cells can no longer be grouped together in the form of tissues or organs. As such, vaccines do not contain parts of fetuses. To manufacture the viruses needed to make vaccines, the cellular suspensions are infected with the virus, and the virus replicates inside the cells. The infected cells break open, releasing many viral particles. Once the vaccine viruses are grown in the cells, the next step in the manufacturing process is to purify the viruses away from the cells and substances used to help cells grow. If you've ever picked raspberries for a pie, you can think of this part of the process as similar. While you're picking, you might get some of the plant, stems, leaves, and even branches, in your bucket, but to use the berries, you remove all of those things so your pie contains only the raspberries. This purification part of the process is important for two reasons. The first, and perhaps most obvious, is the manufacturing reason. From a manufacturer's perspective, an efficient process that results in the purest possible product makes the final product easier to characterize. However, as consumers, the second and more important reason matters more. A pure product will not introduce unnecessary components that could trigger immune responses or affect us in other ways. How can cells from the 1960s still be used today? Cells from decades ago can still be used today because of a process called cell passage, which results in exponential cell growth. So how does cell passage work? When cells replicate in a laboratory setting, they're limited by the amount of nutrients and size of the container in which they're grown. When they reach this point, referred to as confluence, they die unless they're given fresh ingredients and more space. Right before this happens, scientists put 1/5 of the existing cells into 5 new containers. This is called seeding. Over the next few days, the cells in each of those 5 containers will again use the nutrients and grow until they cover the entire bottom of the flask. At this point, the scientists can again do a 1/5 seeding from each of the 5 containers. They will now have 25 new containers. In this way, scientists can make an infinite number of cells. Each time the cells are split into new containers, they are considered to be one passage older. So, in our example, the first split into 5 containers would be passage 1, and the next split into 25 containers would be passage 2. Scientists keep track of this passage number because when cells are passaged too many times, they no longer grow as well. For human cells, including the fetal fibroblast cells Dr. Plotkin used, this number is about 50 passages. This is called the Hayflick limit. So, now you might be wondering, if cells can only reach 50 passages, how do we still have cells to make vaccines almost 50 years later? When scientists are passaging the cells, instead of continuing to make more and more containers, they freeze some. For example, instead of making 25 containers in passage 2, the scientist may decide to make 5 new containers and freeze the cells that would have gone into the other 20 containers. The remaining cells are placed into vials for the freezer. When the cells are frozen, they're stored at about -200 degrees Celsius in liquid nitrogen. At this temperature, biological activity stops. But when the cells are thawed and placed in new containers with nutrients, they again start to grow. Depending upon the cell type and container size they're using, the quantity of cells in this example could produce 8 to 20 vials of cells to freeze. These passage 2 cells can be thawed, typically 1 or 2 vials at a time, to start the cell growth process again at any time the currently growing cells get too old. Likewise, in a few days, the 5 new flasks will grow to confluence, and the same process can be followed, creating more vials to freeze. The important point to understand, though, is that cells grow exponentially, so scientists can grow large quantities of cells to use immediately or to store for future use. In the case of the fetal cells, this process was used to ensure a virtually endless supply. Again, as a reminder, it's important for you to know that any vaccine that relies on these cell lines does not require nor solicit new abortions. Can vaccines made using fetal cells alter a person's DNA? Even though fetal cells are used to grow vaccine viruses, vaccines do not contain these cells or pieces of DNA that are recognizable as human DNA. People can be reassured because when viruses grow in cells, a natural step in the process involves cell death. In most cases, the new viruses burst the cells to be released. Additionally, once the vaccine virus is grown, it's purified so that cellular debris and growth reagents are removed. Lastly, during the process of purification, any remaining cellular DNA is also broken down. What other products use fetal cell lines in development, testing, or manufacturing? In the discussion of vaccination, some have gone so far as to reject vaccines because they could have been developed, tested, or manufactured using fetal cell lines. However, one must also consider that fetal cell lines are not solely used to create vaccines, but are widely used for the creation of life-saving drugs and everyday products. Their use is considered an industry standard in both scientific and medical research. To start, many over-the-counter pain relievers like acetaminophen, ibuprofen, and naproxen have used fetal cell cultures in one way or another. Additionally, many prescription drugs have been tested using fetal cell lines. Even many treatments that have been considered as potential treatments for COVID-19 have been tested in fetal cell cultures. Moving away from modern medicine, food additives and cosmetic ingredients use fetal cell lines as well. The truth is, many of our life-saving drugs and everyday products have been tested on, produced in, or developed using fetal cells. In the discussion of vaccines, if an individual rejects vaccination because it has been developed, tested, or manufactured using fetal cell lines, then this standard should also be applied across the board to all drugs and products. While it's certainly honorable to advocate for not using fetal cells in the production or manufacturing of vaccines, we must remember that vaccines save lives. A number of religious organizations, in their deliberations on this issue, note that the good achieved by preserving or lengthening life outweighs the very remote connection to the once past abortion from which these cell lines are derived and saves millions of lives a year through vaccination. And in reality, as stated by Nicholas Evans, a bioethicist at the University of Massachusetts Lowell, the chances are, if you've had a medical intervention in this country or pretty much any other country, you have benefited from the use of these fetal cell lines in some way. But let's dive into this a little deeper. What are religious leaders' views on vaccines developed using fetal cell lines? The Vatican's Pontifical Academy for Life, the National Catholic Bioethics Center, and leaders from both the Catholic and Southern Baptist churches have determined that vaccines developed using fetal cell lines do not go against the religion's doctrine, and using these vaccines are the best way to protect people from serious vaccine-preventable diseases. The Vatican's Pontifical Academy for Life declared in 2005 and reaffirmed in 2017 that in the absence of alternatives, Catholics could, in good conscience, receive vaccines made using historical human fetal cell lines. The official position, according to the National Catholic Bioethics Center, is that individuals should, when possible, use vaccines not developed with the use of these human cell strains. However, in the case where the only vaccine available against a particular disease was developed using this approach, one is morally free to use the vaccine regardless of its historical association with abortion. The Ethics and Religious Liberty Commission of the Southern Baptist Convention has stated, Christians are not morally culpable if they use treatments and vaccines that were developed using such cells, even if the cells originated in aborted fetal tissue. We hope this video helps you feel more confident in your ability to discuss the use of fetal cell lines in the development and production of vaccines in a meaningful way with your patients. If you'd like more information about this topic, please seek out information from the websites listed on your screen.